for joining the first IAB webinar. Today we will be talking about optical genome mapping. First of all, let me introduce myself. My name is Ondřej Zelba, and I'm the product specialist at IAB, the Institute of Applied Biotechnologies. So who are we? IAB was founded in 2005, and we have a team of roughly 20 employees. And we operate firstly as a laboratory service provider. We have our own lab in Olomouc in Czech Republic. And we also operate as a distributor of instruments, kits, and chemicals. What we like to do, we like to use the technology that we sell in our laboratory, so we can provide the know-how to our customers, and we can also provide troubleshooting when necessary. We focus on solutions for sequencing. We provide sequencing services, mostly whole exome sequencing, whole genome sequencing, RNA-seq, and others. We sequence mostly on our NovaSeq X Plus machine. We also do optical genome mapping services, which we will talk about today. And all of our services are tailored to the needs of our customers. So we approach each and everyone individually and try to find the perfect solution for every customer. As a distributor, we distribute the technology of Bionano, Twist Bioscience, Hamilton Robotics, and others. One of our main goals is to combine technologies from different suppliers. So, for instance, if we want to provide a Twist targeted sequencing workflow, we can also automate it on a Hamilton pipetting machine. Or if we are doing a sequencing service with some difficult samples, we can use a Bionano ionic machine to isolate DNA from the samples and then go on with the service as usual. Again, this is tailored especially for each customer. Since today we will be talking about the optical genome mapping workflow, I should say something about what we do with optical genome mapping. Here is a scheme of the entire workflow. We mostly receive samples from researchers in the field of hemato-oncology, and we mostly use or work with blood samples, fresh or frozen, bone marrow or cultured cells, and even other types of samples. What is necessary to do optical genome mapping? It is first laboratory kits for the sample preparation. Then it's the actual platform, which can be the Stratis system. Then chips are needed. On the chips, the sample is loaded and the chip is inserted into the platform. And then the last part of the process is the bioinformatic part. There is a data analysis that takes place on a special server. After all this is used, then you can visualize your optical maps on a special software. IAB can provide the solution for all these steps either as a service, or we can provide everything necessary for your own laboratory. In our laboratory in Olomouc, it is Regina Bezděková Fillerová who works with the OGM, and she is our main OGM expert. That's why she will be giving the next presentation, and she's also the perfect person to ask more detailed, more technical questions.
So, dear ladies and gentlemen, it's very pleasure for me to introduce this very exciting technology, optical genome mapping. So first of all, I would like to introduce myself. My name is Regina Bezděková Fillerová. I'm application scientist for optical genome mapping technology, and also I'm head of a service lab for this technology in IAB company. Today, I would like to introduce the technology, which is relatively new, and its name is Optical Genome Mapping. This technology is provided from Bionano company, and the IAB company is a distributor of this Bionano. At this moment on the market, you can use the two different instruments for this technology. And let's start and let's think about this technology and ask why we should think about using this technology. Where is the place for OGM on the market and in our laboratories, and especially in clinical laboratories. So on this picture, maybe this picture could help us to answer where is the place for OGM technology. We know that we have a lot of technologies or techniques and method historically for detecting the large structure variation, but still we miss answers for some difficult rearrangements, and we have a lot of problems sometimes to detect some structural variations which are undetectable for some technologies because their resolutions abilities are not enough for that, or we know that we have NGS technology with short reads and long reads, but sometimes it's still not enough. So let's start bringing the gap between cytogenetics techniques and sequencing technologies, and let's go through the technology of OGM. Because maybe it's not something like competition between these main technologies for genomic applications, but these technologies could help us to combine them, combine the methods, and know more and more information about our restrictions and information about our patients. So we know that we have cytogenetics techniques like karyotyping, FISH, and arrays, and we still miss some answers, and we know that the resolution of these techniques it's not enough for us. On the other hand, we have NGS technology and OGM technology, and we think that the combination of these two technologies should be the answer for the whole portfolio of detecting of all structural variations and small single nucleotide variants to copy number variations. So why we couldn't use all the techniques together? And how it works, how we can start with OGM. So first, the most important step is isolation of ultra high molecular weight DNA. So it's very important step. It's not possible to use the classical kits for isolation of DNA. It's very difficult to obtain this high molecular weight DNA, and it's very useful for all next steps because we don't use PCR techniques. So all the time we work with the native DNA fragments, and this is the reason why it's so important to obtain ultra-high long fragments of our DNA. And after this important step, we need to continue with the other steps of this workflow, like labeling with fluorescent colors of these long fragments. After that, we need to linearize these native molecules. We use the specific chips, and we obtain a lot of images of these native fluorescent molecules. And after that, we continue with the analysis and with the softwares, which is very useful for detecting the very complex rearrangements and for visualization of our results. In IAB company, we provide the service which includes in whole steps of OGM workflow. But first of all, we start with the project design, because a lot of customers never meet this technology, and they have a lot of answers about questions about how to start with this project. So which type of sample it's acceptable for this technology. It's possible to use fresh or frozen samples, how they can ship the samples, or if we return the isolated DNA, and so on. So first of all, we start our projects with a discussion with the customers about these questions, and after that, after shipping your samples to our lab, we start with the isolation of ultra-high molecular weight DNA. At this moment, the Bionano company provides different type of kits for isolating of DNA with this type of technology. This technology is named solution phase technology, and it works with the small nanodisk, which is magnetic and covered with a silica membrane. And at this moment, we are able to isolate with these steps DNA fragments, which are long about 700 kilobase pairs. And all questions from the customers are about the type of input material. So first of all, you can use the blood, fresh or frozen, but also we are able to work with the pellets of cells, from bone marrow, for example, from cultivated cells, or with the cells which are cryopreserved. And not at least, we also are able to isolate ultra-high molecular weight DNA from the tissues. So after isolation, we are going through the next step, which is also very important, and this is a direct labeling of DNA fragments. We use the fluorescent labels, and these labels are able to recognize the 6 nucleotides sequence motif. We know the sequence of this motif. And the enzyme is able to attach the fluorescent color to the double-stranded DNA, avoiding the destruction of the DNA fragments. And you can see from this picture that with a new technology from Bionano, we are able to go through this process from isolation to labeling and visualization of molecules during the 3 days. And what is very interesting for us and for our customers, it's to loading of labeled DNA on the chips. How I told, we at this moment on the market provides 2 types of instruments. The older version is a Sapphire instrument, and the new one is a Stratis instrument, which is with a higher capacity. And the big difference between these 2 instruments is that Sapphire instrument use a chip for 3 samples. And the Stratis use the chip only for one sample. And the main advantage of Stratis system is that you can change the position of the samples. You couldn't wait for the second and third sample together to start the run. So you can start each sample individually for the analysis and for the imaging of the sample. But the whole principle of linearization of DNA molecule and visualization through the microscope and laser is totally same for both instruments. And on this picture, you can see the nanochannels, which are the part of this chip. And in these nanochannels, the DNA native labeled DNA go through these nanochannels and is linearized for visualization of the fluorescent labels. And how it looks like, you can see on this picture the images from the camera when the labeled DNA start to go through the chips during the imaging process. And the camera obtained thousands and thousands pictures or images of this native labeled DNA. And after this process, we have to continue with the analysis of the samples and what we are able to detect with this system. So first of all, we are interested about the detection of structural variations in different type and different size. So we can start with a smaller one, deletion, insertions, but also complicated like duplications, inversions, and for sure all of us are interested about detection of translocation. On the other hand, in each run, we are also able to detect copy number variations and newly also loss of heterozygosity. And still, it's not enough for the system. So OGM is able to detect also the complex rearrangements, aneuploidities, also the chromotripsies, and so on. And this ability to detect structural variations starting at 500 base pair. And it depends on coverage of the sample. We are able to detect the structural variations in variant allele frequency of 5%. And if we start with the analysis of our images and of obtaining all the pictures of the native molecules, we have to think about which applications is interested about for us. If we are focused on cancer research and we are interested about detecting somatic changes like in solid tumors or especially in hemato-oncology, or if we are interested about constitutional genetic testing and cell line therapy or gene therapy and QC steps. So at this moment we are interested about germlines. So at this moment, we have to decide which type of pipeline we have to choose. We have two possibilities. If we are interested about germline changes, it means that we are interested about classical genetics. So we need only smaller coverage of our samples. So we are able to obtain enough material for analysis in very short time. And we are able to detect the changes in only in 50% of variant allele fraction. And advantage of this techniques is that we are able to detect the smaller changes or structural variations from 500 base pair. And this type of analysis is named classically de novo assembly, when we are able to construct the whole optical map of our proband and compare this map with the reference genome, and according the changes or differences between the fluorescent labels and their positions, we recognize the structural variations. On the other hand, we also could be interested about somatic changes and about clonality in our samples, which is very often used in hemato-oncology applications. So we choose the second type of pipeline or type of analysis, which name is rare variant analysis, when we cover only some contigs or parts of maps or molecules which are collected together and separately compared with the reference map, and again according the changes or differences between fluorescent labels, we try to find these structural variations. Advantage of this pipeline or type of analysis is that we need the very high coverage, and with a more and more covered genome, we are able to detect or found the variant allele fraction till 5%. With this analysis, we are not able to detect the smaller changes, so we start in 5 kilobase pairs in a limit of detection. And for both type of analysis, we used Bionano Access software. This software is free for all customers, and it's part of this service project. And it's a very nice and very useful tool for visualization of your results in different steps, and also for detecting or filtering your results for your applications. With this software, we are able to detect at the same time structural variations and also copy number variations. But still, it's not enough from Bionano and from this technology, because this year they have the news, and it's the new software, which name is VIA. And this software enables to combine not only OGM results, but combine the results from OGM with NGS results and results from arrays. And this software use the whole big portfolio of filters and classificators, which could help the customers to filter your results, and helps with the interpretation of results and annotation of data, and creating the reports for clinicians. And I would like to also show you the first results or result from our first customers. First example from this project is a constitutional genetic disease. You can see the picture from the patient with neurodevelopmental disease. These results are from classical cytogenetic methods. You can see the karyotype and fish. All both techniques conclude that this patient has translocation on chromosome or between chromosome 3 and 7. And how it looks like with Bionano technology. So first of all, you can see the overview of this rearrangement on the circus plot picture. And you can see at this moment that there are not only translocation, but also some smaller rearrangements which continue with this reconstruction of chromosome 7 and chromosome 3. In detail, you can see circus plot only for chromosome 3 and 7, and you can see that we detect also some rearrangement on chromosome 7, and in more detail with the genome browser, we can see that the translocation between these two chromosomes is also inverted. And on chromosome 7 is inversion of these two parts of this chromosome. And during the reconstruction of the genome of this patient, we also found that this inversion covered the position of gene CNTN4, which contributes to the formation of neural network, and disruption of this gene, which is visible on this picture, can lead to delayed development or autism. And what's new, we found also during this study or during this cooperation with faculty hospital in Brno, we found in one proband where no translocation and complex rearrangements were found. We found a lot of deletions and insertions, and one of them was placed on a position of VDR5 gene, and again we found from the literature that this gene is compared with a regulation of gene expression and affecting again neural development, and dysregulation of this gene again can lead to developmental diseases. The second example of how to use the OGM in maybe in clinics or in some projects in hemato-oncology. It's this project with again our partners from Faculty Hospital in Brno. And you can see the results from multiple myeloma patient when we work or isolate ultra high molecular DNA from bone marrow, from sorted cells separated according CD138 plus marks, and after isolation and processing the OGM, the customer also goes through the classical cytogenetic techniques and from NGS technology. And all of us were very interested about comparison of this several types of techniques which were used for this patient. So we can see the picture of copy number variations detection. So in this part, we can see the results from OGM, and similarly, we can see the same results from classical arrays, from Affymetrix, and also from NGS capture technology. And very, very similar results we obtain also with detection of LOH. So again, you can see the comparison of these three techniques, and you can see that again we obtained almost the same results. What was very, very positive for us. And we were interested also about something new, what we are able to detect with OGM. So in this patient, we found newly this translocation between chromosome 8 and 12. And also not only translocation, but also duplication were detected on this chromosome, on chromosome 8. And at this position, we found the MYC gene. So it's very known proto-oncogene with bad prognostic factor for multiple myeloma. So I could show you several different examples how we use OGM in our projects, but all projects has the same result. So we can see that the optical genome mapping is a very potential new technology which could be for sure combined with the classical cytogenetic method with the NGS. It could help us to found and recognize the variations which are not able to detect with all these technologies. all the time we are able to find something new, and we can see on the newly detected variations in more details, and identified the new candidate genes for different applications. Maybe we can be happy to combine NGS with OGM to obtain the all answers about our genome reconstructions. And the last question is this method at this moment suitable to replace all these technologies and start only with OGM? We thought that at this moment not, but maybe it's the questions for very, very close future. And we recommend all our customers don't trust but verify. So please, if you are interested about this technology, so let's start with cooperation with IAB on your OGM projects. So we are open for OGM services when you can send us only 1, 2, or 3 samples. And part of this OGM service is also a workshop, and all of you are welcome in IAB laboratories, and you can be part of whole processes which are needed for isolation, for labeling, and for analyzing the data from OGM. So at this moment, I welcome you to IAB laboratories, and thank you for your attention.
Thanks for the presentation, Gina. We have a question in the chat. Can you think of a downside of OGM compared to NGS when it comes to throughput or sensitivity/resolution? Also, is it correct that you are able to resolve germline variants excluding SNV, such as indels larger than 500 base pairs or larger structural variants? And for somatic variants, the LOD is in kilobase pair. This is the first question.
We can think that we have to know that it's totally different type of technology, and NGS it's able, the main advantage of NGS it's to know exact the whole sequence of your detected part of genome, so you know exactly or are able to detect the 1 nucleotide changes. With OGM we start on with detection on 500 base pairs. So OGM is not able to detect single nucleotide variants and mutations. So if we are interested about comparison of these two techniques, we have to think about long reads NGS applications. But I spoke about maybe the cooperation of these technologies. So if we start with NGS, so we are able to detect from single nucleotides variants, and if we combine with OGM, so we are able to detect the big and large rearrangements. And the second question was, is there a software solution for co-analysis NGS and OGM data? For sure, the new one, the VIA software, it's exactly for this application, so you can combine NGS data, OGM data, and also the results from your arrays.
Yes, so ideally, if you already have some data from NGS technique or from arrays, maybe you can provide us the samples. We can do OGM, and then you will have a full picture of your samples, full scale resolution, basically. We would like to thank you for your attention and for your participation in our webinar. And we will see you next time. We will let you know when we organize the next part in our series and what it will be about. So thank you and see you again.